there seems to be a single origin and large spread conservation of a single sex chromosome type, but there are lots of other groups, many of the reptiles, amphibians, and fish, where we have multiple origins, turnovers, and loss of sex chromosomes. And it happens so often that we actually can't get a solid count of how often it's happened. And I probably don't need to go into a great deal of detail for most of the audience, but just for the sake of completion, sex chromosomes, the X and the Y, they diverge from each other once recombination between them is halted. And this can have some really nasty consequences for the sex-limited Y or W chromosome, which no longer recombines at all. And so this loss of recombination leads to reduced efficacy of selection and really rapid reduction of gene activity. And many genes on Y and W chromosomes pretty quickly become pseudogenes after recombination is halted. And that's why in many systems, the gene content of the Y or W is much less than that of the X or the Z. And this creates a dosage imbalance. So in an XY system, for any gene that's been lost from Y, that leaves males with one functional copy on the X. And gene expression is not perfectly correlated with gene dose, but gene dose is a pretty good predictor of gene expression. So this reduction in gene dose in males in an XY system leads to, for many genes, a reduction in gene expression, and many genes are actually dosage sensitive. So this can have a pretty nasty phenotypic consequence in males. And as Nicole and others have talked about, dosage compensation mechanisms arise in many systems to try and account for this difference. And the thing to remember, even though the sex chromosomes have evolved many times, is that they're really unnecessary. You can have genetic sex determination with a single SNP. So there's a sex in the pufferfish seems to be determined by a single missense SNP in the gene AMHR2. If you have two functional copies of the gene, you develop as a male. If you have one functional copy and one copy of the missense SNP, you develop as a male. Sorry, if you have two functional copies, you develop as a female. One missense SNP, you develop as a male. So this is sort of in many ways a proto-XY system. And aside from that single SNP, there is no difference between these chromosomes, and there's no evidence of recombination suppression. Now you could argue that this is just a very young system, and there hasn't been time for recombination suppression to evolve. But Nicola Pohm and his group have shown that his hyla tree frogs, they have old but largely undifferentiated sex chromosomes. So sex chromosomes are not a necessary consequence of genotypic sex determination over time. So if it happens a lot, it has nasty consequences, and it's unnecessary, why do we get it at all? And the theory actually dates back to Fisher, as far as I can tell, but was more formally articulated by Jim Bull and Bill Rice and Lee Clark and many others. And the idea is that you have, at some point, the acquisition of a sex-determining gene on what is otherwise a chromosome. So if this is a proto-XY system, you have a sex-determining gene in red. At this point, there's no reason for recombination to be suppressed around that sex-determining gene. That only happens when there's the emergence nearby of a locus that carries sex-specific effects. And at that point, there's selection against recombination. So an individual would always inherit the allele that makes him male with the allele nearby that carries male-specific benefits. At this point, you have the buildup of heterochromatin in the non-recombining region of the Y. Many genes become pseudogenes, and you have the emergence of what we often think of as a strata. And you can have subsequent strata emerging as recombination suppression spreads, until in many cases, recombination suppression engulfs the bulk of the sex chromosome, leaving a very, very small pseudo-autosomal region or PAR at the distal end. So this theory was actually based in part on early observations in guppies by a Danish biologist named Ørjan Binge. And he noticed that a lot of color patterns in guppies read true, so they're passed perfectly from father to son. And Fisher, reading this a couple years later, realized that you can think of color as sexually antagonistic in guppies. If you're brightly colored and you're male, males or females may find you more attractive, so you'll sire more offspring, even though you're diverting carotenoids from other systems in your body, like the immune system, to be orange, and you're more conspicuous to predators. So you may die earlier, you'll have more kids before you do. If you're brightly colored and you're female, there's no fitness benefit at all. Males do not care at all what females look like, but they would suffer all these consequences. And Fisher also realized that Y linkage resolves this conflict, and he predicted that the Y chromosome would in fact become a male supergene. He didn't use the word supergene, that's sort of the modern contrivance of Dobzhansky's co-adapted gene complexes, but he predicted that the Y chromosome would carry quite a lot of male beneficial gene content. Now, I was reading this a couple years ago, I went back and read a lot of the modern synthesis, and I realized that this theory has been driving a lot of what we do in sex chromosome evolution for quite some time, and it's been really difficult to test directly. So we asked the question about 5 years ago now, has sexual conflict over color led to recombination suppression on the guppy sex chromosomes? So we're talking about guppies who live in Trinidad, which is off the coast of Venezuela, and probably around the last glacial maximum, guppies colonized the island. You can see at the north end of the island there's this mountain range, and it's there in particular that they've become really interesting because they've repeatedly moved upstream. There's vastly different ecologies between upstream and downstream populations. And repeatedly and very rapidly, you have the evolution of complex upstream-downstream phenotypes that have sort of spawned a whole industry of work. So people like Dave Resnick and Hood, many others have been studying these different complex phenotypes across these different watersheds. So they're a great model for ecological adaptation. They're also an interesting model for sexual selection. So in many populations, not all, but in many, females have very strong preferences for males with bright colors and long tails. And some studies have suggested that up to maybe even more than 50% of color patterns are Y-linked. So we started this using short read data. We were sort of piggybacking this off of another project. So we used short read data, and we sequenced males and females. And this is a male hyperchromatic species, so we assembled the female genome from our sequence data. And then we mapped those scaffolds back to the reference genome. We didn't want to use the reference genome specifically because it's based on a very inbred lab population that's actually been kept in the lab for over 100 years. So we were worried about biases in mapping, and weird things can happen in inbred populations, but we wanted the positional information that the reference genome gave us. So we mapped our scaffolds back to that, lifted over the positional information and the annotation onto our genome assembly. And onto that, we mapped our male and female reads. So in regions where you have equal coverage, so 2 copies of every chromosome, in both sexes we expect roughly equal coverage between males and females. In regions where the Y has actually degenerated, we expect reduced coverage in males because those Y reads no longer map to the female genome assembly. We are working on fish though, and fish are sort of notorious for having very young sex chromosomes, so we thought we might not see a lot of degeneration of the Y. Instead, we might actually see regions of the Y that are mapping to the X, but have an abundance or an elevated SNP count. So Y-specific SNPs in reads that are mapping to the X will lead to an increase in male-specific SNP density compared to females. So we also looked at that metric. And using this approach, so this is some really excellent work by Allison Wright, we found evidence of 2 strata. And there's some previous work by Christina Dreyer that showed that the sex-determining locus is somewhere at the distal end of guppy chromosome 12. So we're focused on that chromosome. We found evidence of a relatively diverged stratum. So this is older. It's about 4 megabases in total, and it had a significant reduction in coverage between males and females. And there was another stratum, and this is in our lab population, which is a relatively outbred population. So we found evidence of a second stratum of elevated SNP density in males, and that actually was quite a large chunk. It was nearly half the chromosome. And I'm showing you a map at the bottom here. Let's see if I can... Yeah, I don't think I'm clever enough to figure out the pointer. The purple dot is the tail end of chromosome 12, the back end of chromosome 12, and you can see that both in male-female coverage on the x-axis and male-female SNP density is a significant outlier compared to all the autosomes in black and the pseudo-autosomal region, the front half of chromosome 12 in gray. We saw no evidence of decay in gene expression. So I'm showing you a moving average of male-female gene expression, males in blue, females in red. There's no evidence of gene decay on the Y in guppies. And this, at the time, suggested a really rapid spread of recombination suppression. So we were sort of working at this point under the assumption that it was a very young system. And then we next asked the question, does recombination suppression vary across populations as a function of sexual conflict? So we had replicate upstream, downstream samples from 3 rivers, the Yarra, the Kare, and the Aripo. It's actually quite difficult to measure sexual conflict in natural populations, and so we did what many other people have done, which is use the degree of dimorphism, in this case over color, as a measure of resolved, at least partially resolved sexual conflict. So upstream, females have stronger preference for bright male color. As a result, males are brighter. Downstream, females have reduced preference, and as a result, males males are not as vivid. We did a certain amount of population genetic work, just confirming that each of the upstream and downstream populations was monophyletic within the watershed. And we didn't see any difference in stratum 1 whatsoever. So the degree of degradation doesn't seem to vary across these populations. So I'm only showing you here a map of male-female SNP density, and I've highlighted stratum 2 for you in the light purple. And in each of the upstream populations, you can see the degree of male bias in SNP density is higher in the upstream populations compared to the downstream, and this is true when we look at the density as well. I was a little bit worried, and I remain a little bit worried, that people have been moving guppies around Trinidad for quite some time, and if there was one Y haplotype that was advantageous, it might sweep through the population, even if the rest of the genetic material did not sweep through as much. So we did some population genetics at the time, just on sex-linked SNPs, so we couldn't actually identify Y from X-linked SNPs at this point. We saw the same sort of monophyly, so this sort of calmed my concerns a little bit, that this is not sort of a single event that's actually been translocated through human efforts. But this remains a bit of a question at the moment, to be honest. So on the back of this sort of pilot project, we went back and did sample collections in 2016 in Florida, Guyana, and Trinidad. So we went to Florida with Vicki Fuller. We were in Guyana with Godfrey Bourne and Felix Braden, and that's Ben Samcam, and a field assistant, Selwyn France, collecting in Georgetown. And this is Felix in Trinidad exhibiting his remarkable two-net approach to collections. And we wanted to do two things. We wanted to collect related species to date the origin phylogenetically of the sex chromosome system in guppies. And we wanted to recollect the upstream-downstream populations in Trinidad for linked read sequencing using 10x work, so that we could actually get to the haplotype information. So we wanted more samples of higher molecular weight. I'm not going to talk about the second work, because all that we've done at this point is just reconfirm everything I just showed you with more samples and more data. Instead, I'm going to talk about the first bit, where we're trying to phylogenetically date the origin of the sex chromosomes. So this is some work by Yulia, who I think is here, and if you want to see her talk, I'll tell you where to go in the end. She'll do a much better job than I will. So she had 5 species: reticulata, Endler's guppy, Basilia wingii, Basilia picta, the swamp guppy, Latipina, sailfin molly, and Gambusia. And the first question that she had to answer is, how do you assemble and orient scaffolds across these different species without increasing bias? So if we did everything based on the guppy reference genome, there'd be increasing phylogenetic issues as we move further and further out. And so she used a program called Raka with the help of Dennis Larkin, one of the developers. And Raka is actually really sort of amazing because you have both a reference genome, which is an in-group with your target, and an out-group genome, which is out of the entire phylogeny. And so with both those reference genomes, you assemble your genome of your target species, you orient those scaffolds against the reference and the outgroup, and then you use either big pair or linked reads or whatever to pull those together. So this both orients everything and does it in a very sort of even way. So no matter where you are within the target and the outgroup, you have sort of even total phylogenetic distance. So she used this approach, which is what I just said. I should mention that our in-group species was Zygoporus helleri, which is a high-quality genome, and our out-group was our isothides. So I'm showing you here coverage and SNP density, and the regions that deviate from the confidence intervals are highlighted in purple. You can see stratum 1, reticulata, is the difference in coverage, the reduction in male coverage. Stratum 2 is the elevated region of SNP density, and male-female expression. So all of these are log2 male-female expression. Everything is even, orients around 0. And Manfred Charles mentioned quite some time ago that Endler's guppy with wingii probably shares the same sex chromosome system, and we were quite relieved to see that we saw a very similar pattern in wingii. So stratum 1 seems to be largely conserved. Stratum 2 seems to be quite a bit bigger, and there's just a very small par at the distal end of the sex chromosome. What was interesting is that Basilia picta, which is quite diverged, had a difference in male-female coverage on guppy linkage group 12, the same sex chromosomes that you see in guppies and Endler's guppies. And it wasn't a small difference that you can see in the first plot here. There's a major drop in male coverage diverge, suggesting there's large-scale degradation of the Y chromosome. And at this point, we don't really understand what's driving that, if it's large-scale deletions or it's just very rapid divergence. And instead of elevated SNP density in males, because males have lost much of their Y coding content, you see reduced SNP density because they're effectively homozygous or hemizygous for the majority of the chromosome. But interestingly, there doesn't seem to be much difference in expression between males and females. Latipina and Gambusia, there was quite a lot of variance in what was going on, so it's very difficult to know where the sex chromosomes for these species lie. It's possible that it's on the same group, but none of the patterns are really consistent with English developing sex chromosome in either of these species. So this really suggests that the Guppy sex chromosome may be quite a bit older than we'd originally assumed. If it's indeed orthologous with Picta, that suggests it's at least 20 million years old. And what's very strange is that while reticulata and lineage have very little Y divergence, Picta is highly divergent on the Y over the entire length of the chromosome. And yet, there's no evidence of reduced X expression in Picta in males. So Yulia started working with the SNP information that we had from our expression data. There's large-scale reduction in heterozygosity in males. So only 25% of genes on the Y chromosome are heterozygous in males, or sorry, on the X are heterozygous in males. That's compared to 70% across the genome, and that's suggesting that many genes are simply non-functional or no longer expressed on the Y in males. For those genes that are, she did allele-specific expression analysis. And this is just a map here of allele-specific expression for any gene that was heterozygous, that had a SNP, on autosomes in gray, and sex chromosomes in yellow, in males in the first bar, and females in the second column. And you can see in reticulata and Picta, in both males and females on the autosomes and the sex chromosomes, most genes show relatively even expression between the SNPs. The only difference that we see is probably due to cis-regulatory variation. There is a small peak toward the far end for genes that are imprinted, which is interesting from a sexual conflict perspective, but there was no difference in that between the sex chromosomes and the autosomes. In Picta, in contrast, in males, there's a very high peak of strong allele-specific expression on the sex chromosome, but not the autosomes. And this is consistent with large-scale loss or reduction of expression of the Y copy. And yet, when we look at overall expression between males and females, we see no statistical difference on the X in males compared to the autosomes in males, the X in females, or the X, or the autosomes in females. So this is really suggesting that there's a chromosomal level mechanism of dosage compensation in Picta that accounts for this wide-scale loss of Y chromosome gene content. The first case that's ever been known in the TBOS to date. So at that point, we got even more interested in a related species called Basilia picta. This is some work by Ben Sandkamp. You saw him earlier in a picture collecting fish in Georgetown in Guyana. He's particularly interested in this species because it has 5 different types of males. So there's a female, there are 3 male color morphs, the red, orange, and blue. There's a normal pairy male, the one with the stripy back, and then there's a female mimic. These are all very complex morphs that have major differences in physiology and behavior, size, and growth trajectories, and so they're interesting for that reason alone, 5 males. But in addition, these 5 male morphs appear to be entirely Y-linked, so whatever the father is, the sons are. So Ben's actually trying to figure out how the Y chromosome differs across these 5 different morphs, and what sort of balancing selection is actually acting to maintain them all in population. So we see all 5 of these morphs in the trenches in Guyana, which is super cool. But because Picta is a sister taxon to Peri, he immediately started looking at the sex chromosome pattern of degradation. So I'm showing you Picta on the top. This is male-female coverage differences. Peri on the bottom, again, male-female coverage differences. We see exactly the same pattern. Wide-scale degradation of the Y overall, leaving a very small par at the distal end. Dosage compensation to be determined, but you can imagine what I suspect at this point. So, unfortunately, we dated the origin of the sex chromosome. Yulia dated the origin of the sex chromosome, but those left even more questions than we started with. So we seem to have two clades that have a sex chromosome on the same chromosome that's fundamentally different. So we have one clade, reticulata and linii, where we see very little divergence, but sort of elevated SNP density, possibly due to very recent recombination suppression or rare recombination events that sort of keep it from diverging any further. And one other clade, Picta and Peri, where we see large-scale reduction in male coverage, leading to, or related to, essentially degradation or loss of the Y. Why is this divergence so much greater in Picta and Peri compared to reticulata and linii? And I don't know the answer yet. It could be that there's incomplete recombination suppression in reticulata and linii, and that just keeps them from diverging further. It could be that there's independent recombination suppression, these formed independently. It doesn't explain how one diverged so rapidly compared to the other, and this one is much younger than the other. It could be that a large chunk, even most of the Y, has simply been deleted in Picta and Peri. And to answer this, we really need Y chromosome sequence, which we don't have at this point. We have sort of a mishmash of X and Y. And so we've set up families for each of these species. So we're doing transcriptome sequencing on the mom, the dad, and the kids to try and track different SNPs to identify Y sequence from X sequence from autosomal sequence using the sex detector pipeline from David O'Murray's group. So for each species, we have at least 3 families with at least 5 sons and 5 daughters. For Peri, we're setting up at least 3 families for each male morph, so it's quite a lot of tanks. And the hope is here that we will actually have Y sequence from all these different species, and we can phylogenetically figure out if these are actually, if they're ancestral or they're independent events. And for each of these, we're taking it back into the fish. This is a complex tissue, but it also includes the skin, where these color genes are actually expressed. We also don't know how dosage compensation is achieved in Picta, potentially in Peri. And so we're just starting to do ATAC-seq bisulfite sequencing to look at heterochromatin. Chromosome structure is one way you can achieve dosage compensation because it opens up the chromatin to hypertranscribe in the heterogametic sex, as we see in some species. So this is some preliminary work on this by Dave Metzger in the lab. We don't have the Picta data yet. I'm just showing you here, these are regions of before I fall down the stairs. Hypermethylation in males in blue and females in red. And hypermethylation is essentially described as a greater than 20% bias from one sex compared to the other. The gonad is at the top, the muscle is at the bottom. I'm just showing you chromosome 12 here. Muscle is a great somatic control. We don't expect a lot of sex differences in methylation in muscle, and that's indeed what we see. We see lots of differences in the gonad. What's really suspicious is here, right in stratum 1, where we expect the sex-determining gene, we see the highest peak of female-specific methylation that we do across the genome. And this means that there's transcriptional silencing of female expression right where we expect the sex-determining gene to be, which is very suspicious and we're quite excited about. And with that, I'd like to thank the organizers for setting all this up. This has been a really interesting way to think about sex chromosomes, SSE for sponsoring it, and you guys for sticking it out to the bitter end. If you want to hear more about the work that I talked today about, Yulia is giving a talk on Monday. I really encourage you to go see her talk. Jake is also here, and he's been doing this high-resolution characterization of a lot of the color patterns in guppies with the ultimate goal of trying to figure out through pedigrees what is actually Y-linked and what's not, so we can get a much better sense of where the color genes lie. He's talking on Sunday tomorrow. I encourage you to go see his talk as well. Thank the other guys in the lab. I talked a lot about Allison's work, recent collaborators, notably Nicholas Combe. We've been working with him a lot to try and figure out more about the neurogenomics of female preference in guppies. The people can pay us, and if you like the illustrations that I used today of the guppies, that's some work by an illustrator named Clara Lakesey. And her whole goal in life is to create, as she says, biologically accurate but beautifully drafted illustrations. And if you need anything done, I really encourage you to talk to her. She can work remotely. She lives all over the planet. But it was a pleasure to work with her. And I'm happy to recommend her. Thanks again. If you have any questions. There are not plenty of questions. Yeah. We don't know. It's a great question. I don't know yet.
to go over to your discussion.